So many thanks for the invitation to talk about our data, and I'm collaborating, and our lab is collaborating with Fluge and Melle and other people at Haukeland University Hospital. Actually, the oncology department is this part of the hospital, and we have our laboratory at the Department of Biomedicine, close to the hospital. So that facilitates very close collaboration. And what we are after is trying to extract information from samples collected during the clinical trials that Øystein talked about. So doing all kinds of measurements, try to get information, and if possible, try to mimic or recreate some parts of the mechanism in the laboratory, because that would facilitate mechanistic insight, and you can control many factors in the laboratory. So that's our holy grail, to be able to replicate some of the pathomechanism of ME in the laboratory. And I will also start with thanking our collaborators, of course, the people at the hospital and the people in our own lab, and also the other centers involved in the clinical trials, and collaborators that contribute to different parts of the analysis that are done, and also the supporting partners that funds this research. So I will start. I guess you all know quite some about cells, but we are focusing on cell metabolism and cell energy metabolism. And cells, they have a general structure with a cell membrane that encloses the cytoplasm, the inside of the cell. And here you have different structures, different compartments, and they have different tasks and functions. Even though the cells, they are very different depending on the function and their task in the body and the tissue they're in, they have this kind of compartmentalization in the cell. One thing all cells use is ATP, which is the universal energy currency. So all cells depend on ATP, and we have heard about this earlier today as well. And one organelle, which is very important for ATP production, is the mitochondrion. Now, we are focusing on the central pathway of energy metabolism. This is a small window of the huge metabolic map that we have, but it's at the heart of the metabolic map because it produces energy. And it has been studied for many years. We know very much about it. We know that it is connected to many kinds of diseases, and you have changes occurring in this quite restricted metabolic pathway. We have changes there that can be connected to disease. So it starts with the degradation of glucose to pyruvate, and that's the pathway of glycolysis. And you can see there is some ATP coming out there. And then pyruvate is taken up by the mitochondria. And in the mitochondria, that's actually where the combustion is happening. You have a consumption of oxygen, and you have a production of carbon dioxide or CO2. And so during this process, energy is extracted from the molecules, and it is used to produce ATP. And here you have more than 30 ATPs coming from one glucose molecule. Now, this depends on oxygen, and this glycolysis does not. And in the mitochondria, you have the process of oxidative phosphorylation, which we heard about earlier, and also the citric acid cycle, or the Krebs cycle. So this is the basic structure of the pathway. But you not only have sugar or glucose that goes into this pathway, you also have fat. and you have protein. So protein is degraded to amino acids, which can fuel into this pathway at different levels. And then you have fat or fatty acids that can go in and fuel energy production or ATP production. Actually, fatty acids can only fuel the mitochondrial part of the metabolism. And you can see that the amino acids, they are going into different stages here. So the two primary fuels are really carbohydrates and fatty acids. But amino acids are always floating around. The cells does not have a storage place for amino acids, but they are always around, and they are very good fuels if they are around. So we are trying to connect changes in this pathway to pathological conditions. And I will just show you the most classic example of metabolic exhaustion, and that's when you compare aerobic versus anaerobic conditions. So under aerobic conditions, you have oxygen, and then glucose can be degraded completely to carbon dioxide, and you get all these ATPs out of this. So what happens when you do intense exercise? Well, oxygen becomes a limiting factor, and this really shuts down the mitochondrial part. So you lose the ability to extract energy for ATP production here, and then you have to rely on the small amount of ATP you get from glycolysis. But this process needs to be speeded up because it has to compensate for the loss of the mitochondrial ATP. And the pyruvate needs to, you have to get rid of the pyruvate, and that's done by producing lactate. And this is why you can see the increase in blood lactate during this exercise. Now, this is a very classic metabolic pattern that you can easily identify if you see it, and athletes, they're using it all the time to find out when they are at their anaerobic threshold. Now, there are different metabolic patterns that can be recognized and that we know something about, but quite often we also still reaches the limits of our knowledge, and there are still, even though this has been studied for many, many years, there's still new things to learn. And one way to learn about these things is, as we have heard about earlier today, to take blood samples and try to understand what's happening inside the cells by the information you get from the molecules in the blood sample. And typically what you are after, you are looking for fuels and products of these processes. You're looking for signaling substances that can regulate or be involved in regulation, because there is dynamic regulation and there are universal pathways that regulate different parts and the balance between the different parts. And you can also look for indicators of some kind of a depletion of the machinery. It could be some enzyme-related or vitamins or minerals or cofactors, things that are important for the machinery to maintain the activity and support homeostasis. So this is things we are looking for in the blood samples from the rituximab study and also the other clinical studies. And this we published in 2016. And then we looked at the amino acid levels in the blood. And we classified the amino acids into 3 categories, depending on where they enter the central energy pathway. And this was basically the rituximab study, 153 patients. And then we had a control group which was matched with 102 controls. And what we found there was that there was a slight decrease in these 2 categories of amino acids. We call them category 1, 2, and 3. And this was a quite small change, but it was significant, statistically significant, and we think that this indicates some kind of a metabolic shift. It's not a symptom or an indicator of some depletion, or that the patients can just eat more of these amino acids to get better. But this indicates a metabolic shift that tells you maybe that the metabolism is under some kind of stress. Now, we did see this in the female patients. Yeah. So we did see this in the female patients, but not in the men. But there we found other indicators of more compensation. And I will not go in detail, but we think something happens here also with men, but they can compensate more. Now, the stage where we found, if we draw a line between the affected amino acids and the unaffected, we see that we come at the stage where pyruvate is going into the mitochondria, and there you have a key enzyme, which is called pyruvate dehydrogenase. And this is a very complex enzyme, and it's very complex regulated by conserved pathways. And we think this enzyme is important. And we also measured a range of regulators of this enzyme in the patient samples, and we found upregulated several factors that inhibits or downregulate the activity of this enzyme. And this is why we proposed that there is an impaired function of this enzyme in the patients. And what would be the consequence of an impaired function of the pyruvate dehydrogenase enzyme? Well, it works like a valve that can shut, and that means that pyruvate will not enter the mitochondrial oxidation machinery to the same level, but instead be converted to lactate. So that is one consequence. Another consequence is that you will get less ATP, and a third consequence would be that this situation will demand some compensatory use of other fuels, like amino acids, which we measured, probably also fatty acids. So in short, this situation may lead to more lactate, less energy, and the use of backup solution. And we think that this is particularly important under a situation of some kind of strain, if you're doing some kind of exercise. Now, quite often I get the question if and how our results fits with the other results that has been published. And we have heard about some of it today, and several of the people responsible for all the related data are in this room. And if you look at the amino acid composition and the changes in amino acid levels, there is actually, if you go in detail on the data, there is actually quite good agreement. And it suggests that there is a change in fuel consumption based on the reduction in some of the amino acids. If this is true, you would also expect that there are changes in other pathways. And one group has measured the TCA intermediates, that is the Krebs cycle, and they find that there is a proportional shift between pyruvate, increased pyruvate, and reduced amount of TCA cycle intermediates. You will also expect to have changes in lipid metabolism, and several groups have found that. Now, this is part of quite complex data sets with many possible or many different types of findings, but if we go into the parts related to this system, it seems quite consistent. And also, you can take in factors related to redox status. So I would say that there is a quite good agreement that there is a change in fuel consumption based on the analysis in these blood samples. But what does this change mean? I will try to relate this to some other conditions soon. Yeah, here, actually. So, based on the findings in in the blood samples, we try to compare with other situations and what we already know about other situations. And there are similarities between such effects and other well-known contexts, such as starvation and fasting and caloric restriction. And you have hypoxia, training, metabolic stress, and also some other chronic diseases that involves changes in some of these pathways. And, but we don't know at which level the trigger is for causing such a change. It could be a sensor, it could be some downstream process of the sensing mechanism, and it could or could not, may not involve mitochondria. Actually, mitochondria are very sensitive sensor systems of energy homeostasis. So they are likely to be involved at some level, but it's not certain at which level they are involved. And we are trying to, 1 minute. Okay. So we are trying to target these kind of mechanisms in the laboratory, and we are now extending our measurements, doing both focused and targeted and very sensitive measurements, but we also try to go very wide and do global metabolic profiling, as others has done before us. But the aim is to try to identify metabolic patterns that we can link to some mechanistic insight. And one not entirely new theory is that ME involves some kind of a semi-starved condition, or it's similar to starvation in some ways, but not all manners. And typically, you can identify starving or fasting metabolism by measuring some key metabolites. And you can extend these curves as well. But you would expect a reduction in glucose and increase in free fatty acid. And this is because under conditions of fasting, when glucose level in the blood sinks, then you have increased release of fatty acids from the adipose tissue, and this the other cells can use. And you would have also increased amount of ketone bodies under these conditions. And could it be that ME metabolism is kind of stuck in this program somehow? We are trying to replicate some of this in the laboratory, and one way we did this was by exposing cultured cells to serum from patients for 6 days, and then we designed a maximal oxygen uptake test for the cells, used the same instrument as Maureen talked about earlier today, the CRs, and then we can measure lactate and oxygen. And what we did see after exposing the cells to ME serum for 6 days was that actually both lactate production and oxygen consumption increased. And this we think is due to some kind of metabolic stress. The cells need to work harder to survive, and therefore you have a compensatory increase. But we don't know which factor in the blood serum that causes this. It could be IgG, it could be something completely else, we don't know. But there is something in blood that causes this. This is my final slide. So we are linking molecular mechanisms, and we think both the immune system is important and the metabolic system is important, and we are looking at mechanisms that at the interface of these two systems, and we think that this may be the next step. So thank you for your attention.
So my question simply, when do I need to move to Norwich? Can I be part of your trial?
Absolutely.
How do I make it happen?
Yeah, good.
How? What do I do?
Raise some money. For the trial.
How much?
For the trial, we working with the charity, we're raising the money to undertake the trial. We have the timetable. We've already started. So anything you can do to help, greatly appreciated. And we will be hoping to start recruiting patients sort of early next year, hopefully, if everything goes well.
So how do I need to get a referral from my GP?
Yes. There are GP referral forms you can use to refer them to the CSF service at St. Helier. Amalak Banzel there is the the chief consultant. So that's the way to do it. Yeah.
Thank you.
Thank you. Sorry. Yeah.
My question is, in order to avoid the whole glucose process, would a keto diet that's based on fats bypass that?
We don't have a clear answer to that. There are no, so I think there is no, if you have an ongoing fasting process in the body, you would expect that you have also some weight loss. This is some have this, some does not, so it does not seem to be a systemic effect. If you want to kind of have a keto diet to really strengthen your mitochondrial oxidation system, it could be possible, but I really cannot answer if this will work or not.
Hello, thanks. I wanted to ask Dr. Fluge, for the patients that you were treating for cancer, they said their ME symptoms improved. I just wondered whether there was particular elements of their ME that improved. Could you be more specific about
No, at the first time I didn't know anything about ME, so I wasn't so much aware of all the bothersome symptoms the patients had. But I think that after we've seen at least 9 or 10 patients during these years reporting independently recovery, I think it usually affects most of their symptoms. Okay. So I think it's an effect on the underlying mechanisms of ME, not only on the pain or only on the dizziness or some other symptom. So, of course, these are observations. We try to put them into a system to see if this, and you can see the results so far. We haven't any definite answers, so for sure. But the way is we have to move along and try to do the correct thing when we and adjust the course. That's how science is, and yeah. I hope that the main task for us now is to try to understand if the cyclophosphamide track is a real drug track or if it's something else, and also to analyze all the rituximab data and at least the lab samples, trying to see is there a subgroup there that cannot be identified by the usual clinical criteria, but maybe with some kind of laboratory experiment. Maybe, yeah.
That's a not entirely serious question for Dr. Fluge. In view of the quite impressive response to placebo, should we be treating more patients with placebos?
I think it's important to say that in the rituximab trial, we don't have these major, long-lasting, very good responses. Most of the patients had a quite stable symptom course, and there were responders, but we had few dramatic responses in the rituximab trial. What happened in the open-label phase 2 trial, at least? I will probably wonder of that for the rest of my life. But I think it's a mix of natural variation, also placebo mechanisms, and maybe high expectation among patients that can be is known to drive placebo mechanisms, and also possibly some selection. Whether there are patients with a true response to the drug, I can speculate, but I cannot claim that based on the randomized phase 3 trial, because we cannot see any evidence for that in that trial. So, but it will. I know that also other people around the world are still interested in the concept. So we'll have to. We shouldn't put it on the shelf for good until we have analyzed it. And the experiments that David has talked about, and which are extremely interesting, could be a way forward. Also, maybe in the ME/CFS field, to try to elucidate if there are antibodies, and if there are antibodies. Next question is, are they amenable to what kind of treatment? In some autoimmune diseases, you have a success with rituximab, in others you don't. So I think that we have tried plasmapheresis in a few patients. We have tried IVIG in a few patients. That I know that some patients, not in clinical studies formally, but there are ways forward. But I think that what we need now is to pinpoint the mechanisms. and used the device by Ron, looking, is there a serum factor? What kind of molecular weight is it? Is it an antibody? Is it something else? And together I think that Olav told me that when we started this 14 years ago, I had told him, within 1 year we've got it. But and that's for sure not true.
One more question.
It's a question for Dr. Carding. Have you heard of the company and what they're doing with the microbiome in the US? And what's your opinion on it?
Well, we've seen some quite striking changes in the bacteriophage makeup in ME patients, severe ME patients. They're not uniform across all the patients that we've looked at so far, but each has different changes. So we're trying to distinguish the unique features of those and trying to isolate them. So as I said, we're developing protocols for isolating some of these viruses that we can test experimentally, and we are looking at developing maybe an animal model in which we can see if we, for example, introduce these viruses into a mouse that has a human microbiome. Does that cause changes, and is there any physical associated symptoms that might map to ME in those animals? We've considered commercial sources. There's a company called Metabolic as well, which we've looked at. And the advantage they have is they have huge metabolite libraries, reference libraries that we don't have. So it's possible. We haven't pursued it actively. I mean, we are being approached by other microbiome-type venture companies to sort of get involved in some of the analysis. Yeah, and one of those is a metabolome-based one, not the one you've mentioned.
Thank you very much.